All right, guys, let's finish out our biotech notes by talking about something called DNA fingerprinting. So we'll start with what is it? So, DNA fingerprinting is a test where you can compare different samples of DNA to each other and potentially find matches or find commonalities between individuals based on their DNA. To run a DNA fingerprint, you need several different DNA samples, and we're going to watch the process here on the video. But essentially, you're going to do this to all of the samples that you're trying to compare and find some similarities within. So let's check out.
DNA. But to make it understandable, essentially what we have in a human.
Actually, before we do that, let me show you a couple of other things. So the samples that you collect, those samples are going to be sort of chopped up with restriction enzymes. Remember, those are enzymes that are naturally found occurring in bacterial cells, and they make cuts in the DNA. And so these cuts will sort of chop up the DNA samples, and then you're going to put those samples into a really big, very much like what we grew our pea glow bacteria on, agar. You're going to load these samples into an agar gel, and then you're going to pass an electrical current through the gel, and the DNA will start to separate in pieces. So the what I'm calling chunks here, those are fragments of DNA. They will separate based on the size of the fragment, and it will give you this nice little readout of various bands, and you'll be able to compare samples. So when it comes to the DNA that we're using, let's pick back up in this video and talk about what segments of DNA we're actually trying to cut out to run through this agar gel.
Is we have long linear segments of DNA, but within that we have these genes. And so 99.9% of our DNA in everyone is going to be exactly the same. The genes are going to be the same, but you're going to have different copies or alleles of those genes. That's what makes you, you. But if we look into this area in the middle, we used to call this junk DNA, but now we know it's really important in controlling gene expression. We find that there's quite a bit of variability in here, which shouldn't surprise us because the gene makes the protein and the protein makes the phenotype, and that's really what natural selection is selecting for or against. But this in the middle can go crazy, and so it does. And so an example of one that we use in DNA fingerprinting is something called short tandem repeats. Originally, we started with something called VNTR, variable number tandem repeats, and you'll find in DNA sequencing that you have all kinds of. So we had STRs, we have VNTRs. Before that, we had restriction, fragment length, polymorphisms, and so there's a bunch of different things that we could look at. But we're kind of moved to this idea of these short tandem repeats. They work great. There's quite a bit of variability in individuals. And so what is it? You basically have letters of DNA that repeat over and 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 over. So sometimes, 50 times it repeats. And so what does that look like? Well, if we have these three individuals, we'll call this Mr. Blonde, Mrs. Red, and then Mr. Mustache. And so if we look at these three people, their genes are going to be the same, but these STRs are going to be different. These single or short tandem repeats are going to be different. You can see that Mr. Blonde has more than Mr. Mustache and less than Mrs. Red. And so if I make that a little bit easier to grasp onto, if I count them out and then remove everything else, what we get is variability between all individuals. Everything else was the same, but we see variability in here. And we can cut these sections out using restriction enzymes, and then we can amplify them using polymerase chain reaction, and then we can separate them using gel electrophoresis. So how does that work? Basically, I'll take the DNA and I'll put them in a little well. And so we're looking down on this. And so this is a agarose gel. I could put Mrs. Red's and then Mr. Mustache. I could put those all in DNA. Basically, I would then turn on the voltage. So there's going to be a positive charge here, negative charge up here. DNA is a negative charge, and so it's going to be pulled towards the positive. And so what's going to happen is those little fragments of DNA are going to migrate. And so what does that allow me to do? It allows me to tell the difference between each of these individuals. And so this is their fingerprint. But you can tell that this is a really bad fingerprint because we've got these two are exactly the same here. And so when they really do DNA profiling, what they do is they generally use 13 different sections like this. And then those 13 sections are each going to be highly variable. So it's a good way to tell who's who.
Okay, so pausing for just a second, what he has shown us is that the DNA that we're actually sort of taking from our individual samples is not the gene itself, but the spacing in between the genes. Specifically, we're looking for these things called short tandem repeats, STRs. And the reason that we snag those and we want to kind of chop those out and run those in this test is because they show great variation from individual to individual. So, what he was saying is that the gene itself, that's going to be pretty similar within a population based on evolution. But the space in between the genes that really seemingly doesn't do anything, that could change all over the place and not cause a significant impact to the organism. So we see lots of variation in the sequencing between the genes, and that's what we're trying to look at. We're trying to kind of compare samples. We don't want to look at what everybody has that is the same. We want to look at the bits and the pieces of DNA that make us different. And that sort of creates what's called a DNA fingerprint, something that is unique to each individual. And so, what he has showed you here is after cutting out these short tandem repeats, he would load them based on sample. So, Mr. Blonde and Miss Red and Mr. Mustache here would have a sample provided. They would take out the STRs that they want to run for each individual, and they would load them into sort of the gel matrix. And that gel matrix goes into a little chamber, and an electrical current is put through the gel. And we'll talk more about that in just a second. I wanted to point out, though, make sure that you caught what he said about having two STR fragments is not really enough, because chances are some individuals are going to be the same, at least within the length of their STRs. So we want more points of comparison. And so most of the time, like when the police or forensics teams are running these DNA fingerprints, they're looking for at least 13 markers on samples of DNA to be the same before they would really declare with any bit of confidence that the two samples match or are related to each other. So two isn't enough. They would definitely increase how many STR segments that they're going to look at. Okay, let's continue this video.
When would we ever want to do this? Forensics is one reason, and then also in maternity, figuring out who's dad. And so let's talk about the murder. There was a murder that was committed. Somebody was brutally murdered by one of these three suspects. So Mr. Blonde, Mrs. Red, or Mr. Mustache. But they left blood at the scene. And so what I can do is I can grab samples of DNA from each of our suspects, and then I could grab the blood itself, and then I could do DNA fingerprinting on them. So before we separate them, you may think to yourself, which of these looks guilty? Who looks like they're capable of murder? And if we separate them then using that gel, what we can see is that Mr. Blonde is guilty. In other words, his blood matches up with the crime scene. And so what do I mean by matching up? Well, those single or those short tandem repeats, if we look horizontally, are going to be exactly lined up. And if we were to look at Mr. Blonde's son, we'd find more similarities than we would between the others. And so basically that's DNA profiling, DNA fingerprinting. It's much more sophisticated than that, but again, it's kind of on its way out. We'll eventually replace this with DNA sequencing. In the US, the FBI has started creating this database of DNA, which is a little scary. And basically, what they use are 13 different areas within the chromosome or the genome, and then they're looking at those short tandem repeats in there. Now, why do I say that's a little bit scary? I think you really want to protect your DNA, because as we learn more and more about genetics, what's going to be found in your DNA? Well, predisposition to Alzheimer's or breast cancer, any of these things, which your insurance company would love to get a hold of. And so, and it also doesn't answer the idea of Mr. Blonde. Did he really do it? I mean, did the police frame him and then contaminate the blood? So we don't know that. All it does tell us is if we have two samples of DNA The odds of two people having the same DNA fingerprint are astronomical, unless they're identical. But I digress. And I DNA. But to make
Okay, so let's finish up. I think there's one more. Oh, nope, just kidding. Okay, so let's cruise into the next section of our notes. And let's make sure that we understood what STRs really are. So STR stands for short tandem repeats of DNA. And really, these are segments of DNA that sit in between the actual genes. And like Mr. Anderson indicated in his video, the FBI has identified 13 specific STRs that have high variability within the population. And so, because these STR lengths are different from individual to individual, it creates something very similar to a fingerprint, something that is unique, a unique profile to a particular individual. So, we want to compare DNA samples by looking at the parts of the DNA that are unique to us. And so these segments are cut out with a restriction enzyme and they are run through an apparatus right down here in this picture that's called a gel electrophoresis. And so you have a chamber right here. And you will load your DNA into a little gel pad that sits on one of these trays, and the tray gets lowered into the chamber, and you cover it up with a solution, and then you plug this red and this blue electrical cord into something that's almost like a battery, and you turn it on, and then a charge will run through the gel and sort of drag the DNA with it. So let's look at that gel and what I'm talking about. So here, up here is a picture of an agarose gel. And so again, it's very much like the texture of what we grew our pea glow bacteria on in our most recent lab. And there are these little things up here at the top called wells, and they're just indentations. They're little pockets that have been carved out of the gel. And so that's where we're going to actually load our samples from our different individuals. And so you'll load DNA in the well, and the well end of the agarose is placed by the negative charge because the DNA that has been loaded into those wells is negatively charged. So when you flip the switch, the negative DNA will get pulled down towards the positive charge at the opposite end. So apply the charge to the agarose gel. Make sure your negative charge is at the top where the wells are, where the DNA samples are located, and your positive charge is at the bottom. The segments will move through the gel because the negative phosphates on all of the nucleotides are attracted to the positive charge at the bottom. And so the reason that they separate out into different sort of bands is because the small segments move really fast when you flip the charge on, and thus far in the amount of time that you have the charge applied to the chamber. And the large segments, they're really, really big and they're slow, and so they don't move very far. So typically in a gel, the big fragments of DNA will be up here, kind of close to the well. They didn't make it too far. And the little fragments will have made it pretty far down. Now, you can't actually see these unless you've like attached a dye to the DNA sample. And so most of the time, what you're doing is staining this gel pad after you have run your charge for X amount of time. And so it's the staining process that actually makes those bands appear. And so then you get something like this, right? So how could we use these results? Mr. Anderson talked to us about you could compare crime scene DNA to samples from a suspect. So here is a DNA, 6 segments, 6 STRs from a crime scene sample. And here are a couple of suspects. And you can see that suspect number 2 matches with the crime scene sample. And so we could use it in forensics and in criminal investigations. We could also use it in a paternity test, which is a test that figures out sort of who the father is to a particular child. And so what you should know about that is that offsprings, because they come from DNA from mom and dad, they'll have a banding pattern that is sort of a mix from mom and dad. So when you're looking at comparing paternity, you'll look at the child's DNA profile and sometimes mom's profile, and you'll sort of match up the bands that the kid has that came from mom. And then you're left with some extra bands. And so then you could compare those to a couple of possible dad samples and see who matches up with the remaining banding pattern. And that's that last bullet. You can run possible dad's DNA to see if they are sharing banding patterns. So here's an example of that. We would look at the child's DNA, and here's mom. Well, we know that mom gave the child this band and this band and this band, which means that everything else had to come from dad. So I'm looking for a dad that gave the child this band and these two down here. And so you can see dad number 3 is the dad of this child. All right, last thing that I want to talk to you about is something that's relatively new called CRISPR. And so we'll start here with the video of CRISPR, and then we'll backtrack on the notes.
From the smallest single-celled organism to the largest creatures on Earth, every living thing is defined by its genes. The DNA contained in our genes acts like an instruction manual for our cells. 4 building blocks called bases are strung together in precise sequences, which tell the cell how to behave and form the basis for our every trait. But with recent advancements in gene editing tools, scientists can change an organism's fundamental features in record time. They can engineer drought-resistant crops and create apples that don't brown. They might even prevent the spread of infectious outbreaks and develop cures for genetic diseases. CRISPR is the fastest, easiest, and cheapest of the gene editing tools responsible for this new wave of science. But where did this medical marvel come from? How does it work? And what can it do? Surprisingly, CRISPR is actually a natural process that's long functioned as a bacterial immune system. Originally found defending single-cell bacteria and archaea against invading viruses, naturally occurring CRISPR uses 2 main components. The first are short snippets of repetitive DNA sequences called clustered regularly interspaced short palindromic repeats, or simply CRISPRs. The second are Cas, or CRISPR-associated proteins, which chop up DNA like molecular scissors. When a virus invades a bacterium, Cas proteins cut out a segment of the viral DNA to stitch into the bacterium's CRISPR region, capturing a chemical snapshot of the infection. Those viral codes are then copied into short pieces of RNA. This molecule plays many roles in our cells, but in the case of CRISPR, RNA binds to a special protein called Cas9. The resulting complexes act like scouts, latching onto free-floating genetic material and searching for a match to the virus. If the virus invades again, the scout complex recognizes it immediately, and Cas9 swiftly destroys the viral DNA. Lots of bacteria have this type of defense mechanism, but in 2012, scientists figured out how to hijack CRISPR to target not just viral DNA, but any DNA in almost any organism. With the right tools, this viral immune system becomes a precise gene editing tool, which can alter DNA and change specific genes almost as easily as fixing a typo. Here's how it works in the lab. Scientists design a guide RNA to match the gene they want to edit and attach it to Cas9. Like the viral RNA in the CRISPR immune system, the guide RNA directs Cas9 to the target gene, and the protein's molecular scissors snip the DNA. This is the key to CRISPR's power. Just by injecting Cas9 bound to a short piece of custom guide RNA, scientists can edit practically any gene in the genome. Once the DNA is cut, the cell will try to repair it. Typically, proteins called nucleases trim the broken ends and join them back together. But this type of repair process, called non-homologous end joining, is prone to mistakes and can lead to extra or missing bases. The resulting gene is often unusable and turned off. However, if scientists add a separate sequence of template DNA to their CRISPR cocktail, cellular proteins can perform a different DNA repair process called homology-directed repair. This template DNA is used as a blueprint to guide the rebuilding process, repairing a defective gene or even inserting a completely new one. The ability to fix DNA errors means that CRISPR could potentially create new treatments for diseases linked to specific genetic errors like cystic fibrosis or sickle cell anemia. And since it's not limited to humans, the applications are almost endless. CRISPR could create plants that yield larger fruit, mosquitoes that can't transmit malaria, or even reprogram drug-resistant cancer cells. It's also a powerful tool for studying the genome, allowing scientists to watch what happens when genes are turned off or changed within an organism. CRISPR isn't perfect yet. It doesn't always make just the intended changes. And since it's difficult to predict the long-term implications of a CRISPR edit, this technology raises big ethical questions. It's up to us to decide the best course forward as CRISPR leaves single-celled organisms behind and heads into labs, farms, hospitals, and organisms around the world. Wrap your head around the implications of CRISPR with this playlist on genetics.
Okay, so CRISPR is pretty interesting, actually. And they went sort of into detail, but we're going to keep it pretty simple for our understanding. So, CRISPR is a system that was kind of really already at play in bacterial cells. This was part of a bacterial cell's immune system. And you saw in the animation how it worked. So the bacteria got infected with a virus, and that virus was embedded in the DNA of the bacteria. And then the bacteria's immune system, specifically using these Cas proteins, learned that sequence of viral DNA and created a little messenger RNA that was very similar, so that next time the virus was infecting the bacterial cell, the bacteria could sort of destroy that virus. So it basically was trying to figure out what it was looking for upon second exposure, which is exactly what our immune system does. So in nature, when the virus infects the bacteria, the Cas protein inserts viral DNA into the bacterial genome so that that bacteria cell can learn it and create immunity to it. In the lab, they took that naturally occurring system and they sort of edited it. And so they took the idea of how bacteria respond, their immune response, and they created these little guide DNA, or these little guide sequences made of RNA that would be attached to a Cas9 protein. I'm looking at this. This should read RNA. That would attach to a Cas9 protein, and then you would inject this Cas9 protein, and the protein would go find whatever sequence matched that RNA, the guide that you had sort of put on it. And so by doing that, Cas9 would go find a particular gene or a particular sequence of DNA, and it would make some cuts. It would make some edits. And so this entire process is literally a gene editing process. And it's relatively new in the world of science, I would say. In terms of being used like this, it's not new in nature. But you can imagine some of the ethical implications, like the video began to allude to. You can also imagine some of the amazing things that could be done because of CRISPR. So that will wrap up our last section of